So it's a great honor that I have to introduce our speaker, Kyle Myers, Dr. Kyle Myers, for the Marilyn and Jim Simons lecture. Let me just first say a few words about Marilyn and Jim Simons. They co-founded the foundation in 1994 in New York City, and it exists to support basic or discovery-driven scientific research undertaken in the pursuit of understanding phenomena in our world. They've advanced many or funded many advances in frontiers of research in mathematics and the basic science, including, for example, the Simons Observatory in the Chilean Atacama Desert, which will measure cosmic microwave background and will address whether we expand continuously and eventually disappear, or whether we come back, whether there's a expansion and contraction. So the foundation has made contributions to lung cancer screening. They initially funded, after our 1999 article, the Beth Israel Hospital, not where we were, but another program. But eventually we also worked with them. And they funded the open source automated image reading system that we, for a 5-year span, and we're very excited about that. And we've been working with Dr. Kyle Myers on that. So a note about Dr. Kyle Myers: she has her doctorate in optical sciences from the University of Arizona. She's a member of the National Academy of Engineering and a fellow of the International Society of Optics and Photonics, and on the board of, and she was on the board of directors for quite a few years. She's a fellow in the American Institute of Medical and Biological Engineering and Optica. She worked for the FDA Center for Devices and Radiologic Health for many years. Luckily, she's retired from that in order to do other things. And so she's now working on consulting and helping us and other people go through the FDA process with new ideas. So I left out that you were the director of the Division of Imaging Diagnostic Software Reliability. So exactly what we want to do. So Dr. Myers.
Thank you, Claudia. Thank you for having me. Thank you to all of the folks that organized this meeting. It's my real pleasure to be here. So David gave me a really big title in the program some months ago, and I'm going to scope it down a little bit and talk mostly about what Claudia has been teasing you about, which is our response to the need for AI solutions for lung cancer screening. Along the way, I'll certainly touch on where international standards, interoperability can support improved technologies and get us to where we want to go, which is around primary reads. And I feel like all the talks that have come before have really set me up very well. My head is so full of extra things I'd want to say, but I'll try and keep us on time. And I know we'll have lots of time for discussion tonight and through the sessions tomorrow and to come. All right, let's see if we can get them both. So I won't worry about being able to see what's coming next. So as Claudia said, I worked for the FDA for almost 35 years. And so I come to this problem space with a lot of background around what it has taken to get AI to the market over the last many decades. And I want to specifically talk about the particular challenges when we're looking at the problem of screening applications. So my background is broad, diagnostics in general, but I think we all appreciate that there are some special challenges when we're looking to support screening. So to remind you, mammography CAD-E detecting only for breast cancer screening was approved in 1998, approved PMA pathway back when we were scanning film, and Marilyn Geiger in the back of the room here had a very heavy hand in that technology. And so we can talk about what kinds of amounts of data were required to make that happen. 1998 was a banner year for screening technologies for CAD in general. It was our first year that things came through the FDA. And the other thing that was important was automated pap smear systems for cervical cancer screening were also approved in 1998. We have many additional screening applications using imaging on the market that have come through since that time. Those were the ones that created the understanding of what the process might be. We know now, you all know, we have chest x-ray, we have low-dose lung CT for lung cancer screening, we have CT colonography for colon cancer screening, there's optical colonoscopy for screening. And so what I would like to say is, we think for those kinds of technologies, the designs of the studies and the challenges around them are pretty well understood. And I want to especially pay attention to the fact that that 1998 application wasn't just CAD detection, but it was a rule-out system that allowed for the 25% most normal slides to not be needed to be reviewed by a cytologist. The data around that application were published the next year in Lancet. And so let's pay attention to the amount of data that application took. It takes a breath to read the fact that it was 21,700 Pap smears that showed agreement, which wasn't 100%, because nothing will be 100% in agreement with experts, right? But it had similar sensitivity and much better specificity, and moreover showed a productivity benefit. So on the basis of the FDA's review of that data, FDA said, yes, this breakthrough technology, although we didn't have that designation at the time, could come to market. And interestingly, today, that very same approach is marketed by BD through their focal point slide profiler. And pay attention to the fact that in decades, we haven't seen a difference in the operating point. We're still at 25%. And that's because of a number of challenges that we'll all be talking about, right? That have to do with how good the technology can get to be and what are some of the trade-offs with moving that operating point up. So FDA has been very busy authorizing AI/ML applications since the 1998 timeframe where we got things started. And this is data that any of you can find and download in a spreadsheet on the FDA website. What I wanted to point out here are a couple of things. One is that 87% of this data is in the radiology space. It's the highest number of submissions and the steadiest growth rate versus other specialties. And while we talk about de novo applications, a newer technology framework for getting through the FDA, the percentage of de novos is really quite small, about 1 to 3%. They drive 510Ks that can use de novos as predicates afterwards. So I list some of the really important ones in imaging that are new and sexy in the last 5 years, including Quantex, which also comes from Marilyn Geiger's lab, lesion classifier, HeartFlow, a computational imaging technology, triage devices. So we've developed new product codes for each of these kinds of technologies and allowed for the market to grow quite exponentially. And this data is not even being updated by FDA anymore. After the summer of 2023, it's gotten to be so ubiquitous that the website doesn't show any newer applications since that time. So you can see we're just estimating after that. The big point of this slide for you all, though, is for all of that industry, especially in the radiology space, not a single one of these is a rollout system, not a one. Okay, people are thinking about it, working on it. Hasn't happened to date. Some people think about the IDXDR system, which was talked about earlier in the panel. In a similar kind of way, for those of you that aren't familiar with the IDXDR system, because it's outside of lung cancer and radiology, it's an ophthalmic device that was originally passed through the de novo process. So you can see the DEN number means you can look this up as in the de novo database on the FDA website. 18 means it was submitted in 2018. It's a device to evaluate fundus images, and the first application was for a particular fundus camera, not generalizable to other systems until later data came in. One imaging device because of this, challenge that we all appreciate around different imaging devices give you different data, can give you different answers. And this device automatically detects patients with more than mild diabetic retinopathy in patients that are known to have diabetes. So there's a lot of conditions in that, right? You know what your target population is, pretty narrow. And we're going to talk about the data on each of these kinds of applications. And so you can see here, it's a much smaller study, right, than the PAPNet study. 900 patients, 10 sites. They were looking to make sure that this could be a device that would be trained to be used by non-expert users who would take this image and rule in patients that needed further investigation. So one of the ways that FDA looks at these kinds of devices is whether the benefit outweighs the risk. What's the risk? It's that you would have a false negative, a delayed diagnosis. What's one of the ways you mitigate that? Ongoing follow-up. Does that make sense for a lung cancer screening program? Absolutely. But I think, and FDA thinks of this as a rule-in system. So when you think about those distributions of patients with and without the condition, PAPNet moves out people that are on the most negative side of the scale. This one moves in patients that are in the most positive end of the scale. That actually means different trial considerations. And that's important for those of us that are interested in the statistics. So why no rule-out systems in radiology? I think you all know, many of these, that there are unique challenges in radiology relative to non-imaging technologies, and even the imaging ones that we just talked about, the ophthalmic application and the pap smear application, right? Because there's a lot of variability in the normal anatomy in lung cancer, kinds of images and those kinds of data sets. There's variability in the pathology. There's variability in the way that images are collected in terms of different imaging system manufacturers and protocols. And then we have the modern understanding of reader variability, both intra and inter-reader variability. So those distributions here are intended to represent the overlap in the disease cases and the non-disease cases and their scores, and the fact that that's a layering of multiple underlying contributions. And when we talk about standards, what we're really talking about is trying to bring those distributions to a more narrower configuration so that the technology pulls those distributions apart, right? The challenge then, when there's that much variability in the data, today is that clinical studies need to take into account the need to accumulate data that appropriately, adequately represents that richness of that data. And we're challenged by the fact that most data sets aren't diverse enough. They don't represent enough of that variability. So there are two angles we want to go after this with, right? One is get more data, and in the future have standards that are such that we don't need so much data because we've brought down the sources of variability. And then, of course, the cost of annotations and study requirements are high. And so that brings me to this slide and why we're all here, because of this urgent need to address the burden of lung cancer. And this is a slide courtesy of Claudia that you saw in the meeting last year at Verona. And so I'm not going to read it for you, especially the words in bold, because you know this probably better than most of you in the room. But without low-dose CT screening, the bottom line is the cure rate for lung cancer is low. And what we have been seeing and has been talked about earlier today as well, is that a very small fraction of those who are eligible to be screened are being screened. That is, it's just so unfortunate. And so what I'm here to talk about today is one of the things that we're doing to try and address that. And as Claudia has been teasing you with, not only just in my intro, but this morning, is the fact that we have established a project by which we are looking to develop open source software trained and tested on the I-ELCAP data that would be publicly available, intended to support the lung cancer screening enterprise and do this in a pre-competitive way that supports the kinds of innovators that we saw on the panel and those of you in the audience and beyond. So our vision is to develop an open source automated image reading system that we're calling AIRS, that determines that no clinical change in nodules has occurred between a scan today and one taken within 24 months ago. That means no new nodules and no nodule size change in ones that were present in previous scans. So we envision this software as serving as a first read on annual repeat and follow-up CT scans, a first rule out kind of technology. That would mean that radiologists would not need to spend time evaluating lung nodules on those scans, the most time-consuming task. The majority of scans we know in established screening programs are annual repeat and follow-up low-dose CT scans. So radiologists, we understand, would still need to assess for change outside of the lungs. But the rule out benefit, we believe, is quite manifest. We also believe that I-ELCAP has the quantity, diversity, and metadata to bring this to fruition, that data challenge that we've all been talking about that is so important. So here's the envisioned workflow process that our team is looking at. We would imagine a future environment in which participants are selected, those that are eligible. If they pass the selection criteria, they move to a phase where the CT image quality is also assessed. And if that pass is checked, then they move on to software tools that segment the lungs and the lobes, do lung nodule detection, do lung nodule matching between scans taken at different times, and then do a change assessment. And for those nodules in those scans that are passed by the image quality and the patient selection criteria, pass, that data would be determined to not need radiologist review for lung nodules. Any failure at any one of the phases along the way would mean that the scan would need to go for full lung nodule review. We also believe that a tool development effort like this needs an FDA strategy, because we've been talking today about the need for trust around a product. And so our intention is to engage with FDA and leverage what is called the Medical Device Development Tool Program and qualify the tools that are being developed. We're not intending to market them. We're intending to make them open source, give these away, but we want FDA's housekeeping seal of approval in some way. And so what we've identified as our first tack is to go after a medical device development tool qualification, where each software module that you saw in that workflow would be taken to FDA, along with an explanation of the data that was used to develop and to evaluate it and the validation methodology. Now, FDA's qualification program is intended to ensure folks that use an MDDT that as long as you're using the MDDT for the context of use that it was qualified for, that it doesn't need to undergo any further substantive review. The MDDT process is pre-competitive. It's intended to facilitate device development on the part of developers and device review on the part of FDA. And because it's pre-competitive, we believe that it's a great opportunity for you all to collaborate with us, that we can collaborate with developers and future vendors. We intend to share not only the software, but the validation blueprint. How did we evaluate the tools? Our submission documents to the FDA and FDA's feedback. And we believe this will not only support lung cancer screening, but really creates a paradigm that can be copied for other kinds of AI applications in other domains, other modalities, other targets. So we've already submitted our first MDDT application to the FDA last week. So we'll be meeting with them shortly to discuss it. And what's in yellow here is what I'm highlighting denotes the elements that are included in our first submission to FDA. We're calling it nodule match. It takes in CT scans taken within 24 months, segments lungs and lobes. We assume we already have a list of nodules from each of them. Each of those time points and does a matching. So that would be something we'd be giving away, is this ability to make use of our tool to do lung nodule matching between CT scans. You know that the I-ELCAP data represents the largest data set of its kind, including 173,000 scans. We've sequestered data between training and validation on one hand and test on the other, and have already been in the process of ground truthing about 10,000 of those scans. We're taking advantage of the stratified sampling approach that was published by the MIDRIC investigators about a year ago. And Rowena Yip programmed that up to make use of that kind of paradigm to stratify the sampling of the I-ELCAP data in the way that you see here. So you can see the variables that we're considering, which include geography, whether the nodules, nodule status, nodule size, patient age, smoking status, and more. What are we doing about imaging system and protocol differences? Great question. Love to be able to tell you that that's something that's more standard than it is today. That's one of the things that we all need to be working on together. What our approach for now for nodule match is to say, hey, I-ELCAP data represent large numbers of scanners and protocols. The AI is being built on data that represent the diversity of those acquisition systems. We're limiting the data to slice thicknesses of 1.25 millimeters or less, but across all recon kernels. We know more standardization and QA/QC would reduce variability in the data, and Rick Avila is such a leader in this, is going to talk some more tomorrow about how we could do better in the future and how we might harmonize data from the past. And that we really believe this community can work together to help to support the recommendation development for more consistent acquisition settings. So variation in the data contribution can be reduced so that our tools can focus on biological variability. So MDDT qualification is something FDA has been very strongly saying these tools can and would be welcome to be introduced through collaboration in non-competitive settings through the formation of consortia or other collaborations to foster MDDT development. And so we're really excited about the possibility of growing what we're doing with you all. I wanted to say something that hasn't been mentioned yet around tool evaluation in the presence of a rule-out system. Because this is an area in which there are new statistical tools that are going to be needed. That the standard approach for FDA to evaluate an AI algorithm has been to say, tell us what the ROC curve is for the computer alone. Even if the operating point of a computer is going to be eventually fixed when it's doing its support to a reader, we ask for standalone performance across the board. Then we would also say, tell us what the reader does without CAD alone. Why? Because that is essentially a way of telling FDA how difficult the data set is. Because we're not very good at being able to say in advance, do you have representative data, or have you chosen the most easy cases, the most normal and the most abnormal? And then finally, how does the reader do in the presence and with the support of the tool? So standard FDA evaluation uses reader alone performance to assess case mix difficulty. AI standalone performance is useful, especially when you want to come back to FDA with a new and improved tool. And then reader plus AI shows readers' ability to benefit, which actually is kind of interesting in this slide, because you can see the readers benefit down here, not so much up here. Maybe that's training, maybe that's something else going on, not the point of this talk. The challenge is that that's a really different paradigm from taking cases out of the mix. So now we're not going to have readers alone and readers plus CAD and an ROC curve for that. We're going to have readers without CAD, and we're going to have CAD without readers. And there isn't really a consensus yet on how you make a metric for that, or even an ROC curve. So here's a little toy problem. Suppose you had a distribution of cases radiologist alone. So you got the positive cases are here, the negative here, always some overlap. There's the ROC curve. What happens if I rule out the most normal cases? Okay, what happens to the ROC curve for the readers, and what am I going to tell you about the data? Well, it's actually kind of interesting, and this is going to always happen, is that the reader ROC curve is going to move to the right because those are probability distributions. So you have changed the case mix. You made it harder for the readers, okay? And so it's the case that moving the most negative cases out of the box drives down reader performance. This is not unexpected, and we're going to see publications around this for people that are going to be only looking at the readers, and we're going to say, yep, we knew that was going to happen. You need to account for the fact that, you know, the cases that aren't in this mix anymore that were handled by the AI and how well that was done. So how do we assess the impact on classification performance when a case is seen by a reader or an AI, but not both, is an open question. As I said, the case mix will be more difficult for readers. The sensitivity of readers alone will be non-superior. Specificity must be non-inferior. FDA has been working on this. The prevalence of disease will be higher in the cases that are left in the reader's box, and that will likely, especially in the short term, move readers' operating points. So there's some really interesting study designs that need to be considered and figured out as we talk about where are standards needed. Now, FDA has never liked sensitivity specificity pair comparisons, and they're definitely not going to like it here. So FDA just put out on the archive a recent paper that I've got down here, investigating some other metrics, including what they're calling the equivalent relative utility. This is going to be a cost-benefit evaluation that we're all going to need to put our best statisticians around working together on, another pre-competitive challenge. So there are many data and AI evaluation models that are emerging. We heard a great talk from Marilyn Geiger on MIDRC. Many of you are probably aware of the medical imaging data marketplace concept that was just presented by ARPA-H as a collaboration with FDA as another approach to getting data owners to share with AI developers. And then a number of other logos here of folks that are either saying, give me your data, or I'll help you validate your algorithms. So why are we taking the approach that we're taking? What's the value proposition for I-ELCAP, open software tools? Well, I list them here. AI developers, while we're not sharing the data today, we are sharing software that will represent the data. The software represents the largest and most diverse curated data set that is longitudinal. You would see open source, that gives you the ability to use it out of the box or to make adjustments to it. You can put a wrapper around it. You can do what you want. But we know that having made that available to you accelerates development time and review time. That for healthcare partners to I-ELCAP, this ensures that exposure to their data is being, avoided while taking advantage of the fact that the data is diverse, presenting products to healthcare providers that are derived from that data. Patients' data is protected while they're able to know that their data is being used for public good, and that even patients outside of the I-ELCAP system will eventually benefit from these products. For FDA, the reason that we love MDDTs and this kind of approach is that it prepares the agency to review tools that were developed from it, builds awareness of this kind of process because we're hoping that others will follow our lead. This is really not about imaging, but I just love this quote: Americans deserve accurate and reliable diagnostic tests. And that's what we're about. And Jeff Shuren, who is the longest tenured CDRH director, Center for Devices and Radiological Health at FDA, is very interested in hearing in the process of review, not just from device innovators who've submitted products to FDA and said, my device is good enough, but there's an incredible room for technology developers and providers to jointly talk about what they believe is good enough. And now they have an approach for getting patient voices as well. And that is because we're very interested at the FDA in putting patients first, and that includes patient perspectives. And I think that this quote is especially also relevant when we're talking about a global data set and a global community as represented here today. So in conclusion, rule out systems in radiology don't exist today. We're looking to develop one that would facilitate lung cancer screening, making use of open source software tools qualified by FDA, sharing the software and sharing our experience with FDA to speed innovation in this space and others. I'm looking for people to come together to develop consensus around many different elements of the workflow in terms of study design and endpoints and acquisition guidelines to narrow down the kind of data that would need to be evaluated and things related to metrics. So with that, I especially want to thank the folks on the AIRS team. The smallest cog in this really big wheel with our visionaries, Claudia Henschke and David Yankelevitz. Thanks for inviting me to be part of your vision. Rick Avila, who leads the AIRS AI algorithm development team. Rowena is doing all the statistics, and unfortunately, Artit couldn't be here, our lead engineer, who's home taking care of a new little one, so we wish him well. So thank you for that. I'll leave you with this, and happy to take any questions.